Hi, I am Rasheda Mim. I did my research on comparative analysis of protective effects of spirulina extracts against lead-induced neurotoxicity. Let's first understand the reason behind this whole project. When lead enters the cell, it interferes with normal cellular processes. During normal metabolism, especially in the mitochondria, small amounts of reactive oxygen species, or in short ROS, are naturally produced. However, in the presence of lead, this balance is disrupted. Lead increases the production of ROS and also weakens the cell's antioxidant defense system. These ROS are highly reactive molecules that can damage proteins, lipids, and DNA. Uh, this, led, this leads to oxidative stress, which reduces cell function and can eventually cause cell death. This is especially harmful for neuronal cells as they are very sensitive to oxidative damage. So, finding a way to protect these cells is the main motivation behind this study. To address the problem, we looked for a natural compound that can reduce oxidative stress. One promising option is Spirulina plantaceus, a type of microalgae. Spirulina is rich in proteins, phenolic compounds, and antioxidants, which can help neutralize reactive oxygen species and protect cells from damage. For this experiment, we used sci-fi cells, which are commonly used as an in vitro model to study neurotoxicity. Based on this, we hypothesized that spirulina extracts would reduce lead-induced toxicity in sci-fi cells with a difference in effects between aqueous and ethanolic extracts due to their distinct chemical compositions. The objective of this study was to extract bioactive compounds using aqueous and ethanolic solvents, characterize extracts, evaluate protection against lead-induced toxicity. Let's move to our method section. First, we perform extraction using water and ethanol. After extraction, the next important step was to understand the chemical composition of each extract. And for this, we perform BCA assay to determine protein content, FC assay to measure total phenolic content, DPPH assay to evaluate antioxidant activities. In addition, UPLC was carried out to identify and compare bioactive compounds in both extracts. For this, we use 4 standards, namely gallic acid, caffeic acid, p-cumaric acid, and quercetin. Then we perform our biological study using sci-fi cell. Different dilution of spirulina extracts and varying concentrations of lead were first tested individually on cells. Based on these results, one optimal dose from each treatment was selected. These selected doses were then used for the combined treatment, where cells were exposed to lead along with each extract to evaluate their protective effects. All treatments were carried out for 48 hours. Cell viability was then measured using the MTT assay. For statistic analysis, t-test was used for chemical assay and one-way ANOVA was used for cell study. Here is our outcomes of all experiments. From figure 1, if I zoom a little bit, from figure 1, it can be seen that aqueous extract showed more compound peaks, including all 4 standards here. And in contrast, the ethanolic extract exhibited fewer peaks and only 2 standards, caffeic acid and quercetin. This result supports the chemical assays in figure 2. where aqueous extract showed significantly higher protein content, total phenolic content, and antioxidant activity. And here is our biological studies outcome. Figure 4a showing like in figure in this in the spirulina extract test, it is clearly shown that compared to the vehicle control, the eightfold dilution of each extract showed significantly increased in cell viability. On the other hand, in the metal study, compared to the negative control, uh, 25 micromolar concentration of lead showed a significant decrease in cell viability, but did not cause complete cell death, which when in in figure C, when combined spirulina, when uh, metal with spirulina extract was tested, cell viability improved compared to the lead alone. It is also noticeable that among the two extracts, the aqueous extract showed stronger protective effects than ethanolic extract. Here, this is our result, and overall, this study's findings showed that both both type of extract has potential to give protection, and overall this study's finding actually showed that both type of extracts has potential to give protection against metal, even though the aqueous portion has more compounds and protective ability compared to ethanolic extract. However, higher doses of spirulina showed some potential toxicity, while lower concentration were safer, indicating a dose dependent effect. Future work is needed to optimize the extraction method, identify the active compounds, and test different doses, exposure time, and cell models to better understand the mechanism and to confirm these findings. Here is my references. I would like to sincerely thank my supervisor, Dr. Jessica Livingstone, for her guidance and support. I also thank Mr. Michael Gibson, Marla McKenzie, Crystal, and Shanda for their lab assistance.